Good morning and welcome to all. So today we are going to discuss the new unit, that is classification of microorganisms. So according to the microorganisms, we know that we have studied in the previous class the organisms which are not capable to see with our naked eye. We use microscopes for observing microorganisms. So these microorganisms are classified into taxonomic categories to facilitate the research and communication. So for the further study, we should classify those microorganisms in various taxonomic categories. So life on the earth is famous for its diversity, means it has different types of form. Microorganisms not only available in a single form, it can be available in various different forms. Throughout the world, we can find millions and millions of different forms of life. And the biological classification, it helps to identify each form according to the common properties or similarities using a set of rules and an estimate as to how closely it is to a common ancestor in a way to create an order. So this biological classification helps to estimate or closely relate them to a common ancestor. So from a common ancestor, different forms have been erased. By learning to recognize certain patterns and classify them into specific groups, so these biologists are better able to understand the relationship that exists among variety of living forms that inhabit the planet. So these classification methods help to understand the different types of forms which inhabit the planet. The first, largest and most inclusive group under which these microorganisms are classified is called domain. I think you are familiar with these terms in the +2 when we are studying classification. So domain is the most inclusive group and it has 3 subgroups under the domain: bacteria, archaea and eukarya. So the first group defines whether an organism is prokaryote or a eukaryote. So the first group which helps to define whether the organism is either prokaryote or eukaryote. And this domain was proposed by the microbiologist and physicist Carl Woese in 1978. And it is based on identifying similarities in ribosomal RNA sequence of microorganisms. So based on the similarities in the ribosomal rRNA sequence within the microorganisms, organisms can be classified into 3 subgroups. The second largest group is called phylum. So first one is domain and the second one is kingdom. Major kingdoms have been described and it includes Prokaryota, Protoctista. That Prokaryota includes archaea and bacteria. Protoctista includes protozoa and algae. Then fungi, plantae and animalia. And this kingdom, it is further split into phylum or division. Again this kingdom is split into phylum or division, then class, order, family, genus and species, which is the smallest group. The science of classifying this organism, that is termed as taxonomy. So taxonomy is science of classifying organisms and the groups making up this classification hierarchy. They are called taxa. I will show the taxonomy and taxa in later slides. So the taxonomy, it consists of classifying new organisms or reclassifying existing ones. So it helps to classify new organisms or reclassify existing ones. The microorganisms are scientifically recognized using binomial nomenclature. So we can recognize, we are classified using taxonomy. Then we are recognized, this scientifically they are recognized using binomial nomenclature. That is, using this we can name the microorganisms using two words that are genus and species. Now, naming of the microorganisms binomial nomenclature. Two terms which turn microorganisms name genus and species. The names assigned to the microorganisms are always in Latin. So we are naming the organisms in the Latin language. The first letter of the genus, it should be always in capital letter. Then the genus, first of all genus, then species. Genus appurum capital letter aayirikkanam. Species always in small letter. The classification of microorganism has been largely aided by the study of, so main ആയിട്ട് microorganisms classify ചെയ്യുന്നത് അവയുടെ fossil study and also their DNA sequencing വഴിയാണ്. We are classifying each and each microorganisms into various groups. Now method of the classifications are always changing. The most widely employed methods for classifying these microbes are, first of all, morphological characteristics, that is external appearance. Then differential staining. While we are staining, we can observe different pattern. Then biochemical testing. That means what are the different types of biochemical reactions that takes place in each microorganisms. Based on that, we are classifying microorganisms. Next is DNA fingerprinting or DNA base composition. That are highly advanced methods for classifying. Then PCR, polymerase chain reaction. Then DNA chips. So this DNA fingerprinting, DNA base composition, this PCR, DNA chips, ഇതെല്ലാം advanced methods ആണ് ട്ടോ classify ചെയ്യാനുള്ള. Now what are the historical challenges of this classification? Despite there being little agreement on the major subgroups of the bacteria. അപ്പോൾ ഈ classification scheme നേരത്തെ പറഞ്ഞു, but there was a little disagreement in this. അതായത് classification system ഇതിന്റെ ബേസിലാണോ എന്നുള്ളതിന്റെ ഒരു ആർഗ്യുമെന്റ്സ് നടന്നിരുന്നു on the basis of the subgroups. So finally gram staining results were commonly used as classification tool. Then അതിൽ തന്നെ ഇപ്പോൾ നമ്മൾ സെക്കൻഡ് പറഞ്ഞത് ഡിഫറൻഷ്യൽ സ്റ്റെയിനിങ് ആണല്ലോ. അപ്പം അതിൽ ഗ്രാം സ്റ്റെയിനിങ് തന്നെ അതിൽ വരുന്നതാണ് ഈ ഗ്രാം സ്റ്റെയിനിങ് എന്ന് പറഞ്ഞ മെത്തേഡ്. അപ്പോൾ അത് കോമൺ ആയിട്ട് ക്ലാസിഫിക്കേഷൻ ടൂളിൽ എടുത്തിരുന്നു. ഫോർ എക്സാമ്പിൾ, the prokaryotes share many common ancestors such as lack of nuclear membrane, unicellularity, that is all of the prokaryotes are unicellular organisms and their division is by binary fission, and also they are generally of small size. So based on this, gram staining can be commonly used as the classification tool to identify the bacteria. Until the advent of molecular phylogeny, the kingdom Prokaryote was divided into 4 divisions. That is a classification scheme. It was still formally followed by the Burgess Manual of Systematic Bacteriology. So we are following this Burgess Manual for classifying the bacteria. Next is the various species among each other based on the several characteristics which are determined by the gram staining, which allowed their identification and classification. So when there are different types of species present, so they differ. Now what are the major groups of this system? Which means that Major groups of the kingdom Prokaryote, which was defined by Burgess' Manual of Systematic Bacteriology, that is Gracilicules, which means gram-negative, Firmicutes, that is gram-positive, molecules that are gram-variable or example mycoplasma, then mentocutes. These are uneven gram strain, that is methylinogenic bacteria now known as RK. So these are the four major subgroups coming under the kingdom Prokaryote. Okay. Now next is the molecular classification. In the molecular era of classification, Carl Woese, who was regarded as the forerunner of this molecular phylogeny revolution, he argued that the bacteria, archaea and eukaryotes represent separate lines of descent that diverged early on from an ancestral colony. So he was arguing that bacteria, archaea and eukaryotes are separate lines, but early they have arisen from common ancestral colony. The few biologists argued that, so this was the argument of Carl Woese. So there are another biologists. They also argued that archaea and eukaryota arose from a group of bacteria. In any case, it is thought that viruses and archaea began relationships approximately 2 billion years ago, and co-evolution may have been occurring between members of these groups. So there are different types of arguments. It is possible that the last common ancestor of the bacteria and the archaea was thermophile. Thermophile means those forms which exist at higher temperature. And these thermophiles raise the possibility that lower temperature also it is an extreme environment. So this thermophile lives in extreme environments such as higher temperature. Same to these thermophiles, this lower temperature, that is cold conditions, they are also extreme environments. So in archaeal terms, and organisms that live in the cooler environments appeared only later. So first we are classifying using these thermophiles. Since the archaea and bacteria are no more related to each other than they are to eukaryotes. So the term prokaryotes only surviving meaning is not a eukaryote. So similarities exist between archaea and bacteria but not with the eukaryotes. With improved methodologies, it became clear that methanogenic bacteria were profoundly different and were erroneously believed to release that of ancient bacteria. Methanogenic bacteria are what? Coming under archaea. Thus, though Woese identified that the three primary lines of descent, the archaeobacteria, eubacteria and eukaryotes, the later now represented by the nucleocytoplasmic component of eukaryotes. So these lineages were formalized into rank domain and which divided life into three domains. So finally, the domain, there are three domains: eukaryota, archaea and bacteria. This is the final lineage. So this scheme is still followed today. In 1987, Carl Woese divided the eubacteria into 11 divisions based on 16 rRNA ribosomal sequence, which were several additions are still used today. So this is the main classification scheme of this bacteria, and in this it comes most of the organisms. So further classification of domains are as follows: mainly domain, kingdom, phyla, class, order, family, genus and species. These are the main classification domain. The binomial nomenclature assign each microbes in two terms: genus, always capitalized, and species, always in the lowercase letter.